Okay, so our goal in this project was to better understand the diversities of fruit flies in the Eugene, Oregon area. The species distribution of species populations shifts in accordance with climate change, invasions, and other biotic and abiotic factors. The genus Drosophila, with thousands of species, is particularly notable for changes in species distribution over the course of human history. The native species of fruit flies in Oregon is Drosophila melanogaster. However, the extent to which invasive species has impacted fruit fly populations in Eugene, Oregon remains unknown. Our class chose to conduct a survey of the fly population present in Eugene. This allows us to better understand the present biodiversity of fruit fly species in Eugene, Oregon.
The goal of this experiment was to understand the biodiversity of the fruit fly population present in Eugene, Oregon. Our research was guided by the question, how does biodiversity of different fruit flies vary in the Eugene, Oregon area?
To do this, we collected 85 wild fruit flies using fermented bananas, soda bottles, and fly vials. First, we cut a small slit into a 2-liter soda bottle. Then we placed a banana yeast mixture at the bottom of the soda bottle. Leaving the bottle outside, the flies were able to enter the bottle through the slit. After 3 days, we inserted the fly vial at the top of the bottle, and using a trash bag, we covered up the bottle, which allowed for the flies to fly upwards and enter the fly vial where they were collected. We then extracted DNA, amplified a fragment of the 16S gene using PCR, and sequenced this fragment using Sanger sequencing.
After receiving the DNA results, we compared the nucleotide sequences to public database using the BLAST software to identify the species. As you can see over here, these figures are the bottle trap that was used, described by Hannah, and this figure over here is the BLAST sequence, where the BLAST software where we inputted the sequence to compare the data.
All of our collected flies that were sequenced were identified as Drosophila suzukii, which is an invasive fruit fly species. So none of the 85 flies were identified as the native species Drosophila melanogaster. So figure 3 shows a DNA chromatogram that provides us with the results from the initial Sanger sequencing. And figure 4 is an example of one of our BLAST results for a specific fly sequence. And the result shows that the specific nucleotide sequence matched with an already known Drosophila suzukii sequence.
In conclusion, our findings showed how invasive species have caused a lack of diversity in Eugene, Oregon's fly population. As seen in the results section, the invasive Drosophila suzukii species has far surpassed that of the native Drosophila melanogaster population. These concerning findings reiterate the importance of controlling the Drosophila suzukii species to avoid not only a loss of diversity, but also an extinction in our native fly population.
The results showed that 100% of fruit flies caught were Drosophila suzukii. Drosophila suzukii has outcompeted Drosophila melanogaster, and these findings reiterate the importance of controlling the Drosophila suzukii population and rehabilitating Drosophila melanogaster populations.
We'd like to thank Annette Esteves for performing PCR amplification and DNA sequencing for this project. Also, thank you Nadia Singh for leading the SCORE program and advocating for diversity in research. This concludes the undergraduate research symposium presentation on the lack of diversity in Drosophila species in Eugene, Oregon. Thank you for listening.